All right, people, this is the lecture on polypeptide synthesis, also known as protein synthesis. We'll go into why there's a slight difference in those. More importantly, this is the central dogma of biology, the most important concept, because this is what takes us from DNA to RNA to protein, and then the expression of the protein is what builds molecule cells, which then ultimately gives us our phenotypes and the physical aspects of who we are. So here we go. So we know that DNA is the control molecule of the cell, but really DNA doesn't do anything except give directions. So, and that is by making proteins through the first synthesis of RNA. So what we see in this picture, we see our DNA up here that is being copied by RNA polymerase into our RNA. And then the RNA goes out into our cytoplasm, works with the ribosome, with tRNAs to bring in all of these amino acids to build this polypeptide chain right here. And of course, because it's a protein, it's going to fold from the primary structure to the secondary structure to the tertiary and quaternary structure, and then the structure of that protein will determine its function. So very, very, very indirectly, DNA controls the cell by making these proteins which do a specific job. All right, so originally this concept was called one gene, one enzyme. Once scientists figured out that DNA was the molecule of heredity and all the history stuff that we talked about previously, then there were some Beadle and Tatum, two scientists. They were working with mold, very exciting. And what they were doing, they were growing the mold in different cultures, and they noticed that certain mutant molds could not grow unless they were given a supplemental amino acid. These wild type molds could grow on just bare minimal nutrients because they had all of the correct genes to make all of the correct amino acids to build their proteins, but the mutants had a mutation in the DNA which prevented a correct enzyme from being formed to make a specific amino acid, and then they couldn't grow unless that particular amino acid was present. So, so these different class 1 mutants, what we see in this first one, they couldn't, or down here we can see that they needed to have ornithine amino acid given to them. These needed citrulline, and this one needed arginine. And so what the scientists figured out from this is each gene that was mutated made one enzyme which gave one particular function. So the first idea was one gene equals one enzyme, but then scientists learned that not all proteins are enzymes, so then they clarified it to one gene, one protein, and then as scientists learned the different levels and structure of proteins, they came up with the idea that, well, if you look at something like hemoglobin, it's made up of 4 subunits of 2 different kinds of proteins. So then it became one gene, one polypeptide, and then those polypeptides formed together to give you a quaternary structure. So then it wasn't good enough to say one gene, one protein. Now it's one gene, one polypeptide, which is why it's called polypeptide synthesis instead of protein synthesis. That's that minor clarification. Does it really matter? It does not. But we will also learn later when we get into genomic control that one gene, one polypeptide isn't quite accurate anymore either, because RNA can be edited in different ways so that one gene can actually give you multiple polypeptides. So still figuring stuff out. Okay, so from gene to protein, 3 basic steps. Transcription, that's where the DNA gets copied to RNA. Then if it is messenger RNA, it will get processed in 2 main ways. And that will then give us functional mRNA as our base codon, not code because that's the DNA, to make the protein. And that protein is made in translation, and that's the big building process. All right, so here we have a gene. This gene, DNA, is made into the RNA, and then the RNA is made into our polypeptide chain. Of course, polypeptides are going to be hundreds to thousands and thousands and thousands of amino acids long, not just 4. But what we want to see here, again, back to our directional idea, we see that our RNA, the 5' to the 3' end. So this is how the RNA is built in this direction. In order to do that, it is built anti-parallel to the DNA. So the DNA is read 3 to 5, while the RNA is being built 5 to 3. And then what we see, every 3 RNA segments, every 3 RNA nucleotides, is going to be used to dictate what amino acid is brought in. So this is called a codon, or a codon triplet, because it's 3 nucleotides, and each one brings in an amino acid. And we'll learn how to interpret the RNA into the amino acids later. All right, so RNA, so typically you've heard of mRNA, tRNA, and maybe ribosomal RNA, but there's actually a whole bunch of different ones. The 3 main ones, messenger RNA, that's the copy of the DNA. Then the transfer RNA, or the tRNA, and then the ribosomal RNA. These are the main ones that are responsible for synthesizing the polypeptide, but there are some other ones. We have small nuclear RNA. So this is going to be part of what's known as a SNRP, or a small nucleoribonuclear protein, which is something that makes up a cluster of proteins called a spliceosome, and that's what helps edit RNA. Then we have signal recognition particle RNA. This is going to be what is responsible for determining whether a protein stays in the cytoplasm or if it is going to be made in the rough endoplasmic reticulum. So what we see here, you have a ribosome. It is attached to an mRNA. It is making a protein. And then if that protein has a specific sequence of amino acids at the beginning, this is called a signal peptide, that will bind to that signal recognition particle mRNA. And that brings it down, binds it to a protein on the membrane of the rough endoplasmic reticulum. That's what this blue stuff is here. That opens up a pore, and then that protein gets made and enters into the rough ER. So then this end piece right here, that will pinch off and make a vesicle, and that will go to the Golgi, where it'll get modified and shipped and whatever into another vesicle, and then that will go out of the cell at the cell membrane, thinking back to our backpacks in the parking lot example. Other RNAs that are involved, so this small nuclear RNA helps make ribosomes. That one's not super important. Small interfering and microRNAs, these are important for what is known as post-transcriptional gene expression control. So after RNA is made from the DNA, pausing for the automatic cat feeder, these two things can bind to the RNA to block translation and prevent the protein from being built. So what we see here, we've got some RNA that gets cut up by something called a dicer molecule over here. And then we can see here that these small pieces of RNA bind to the mRNA, and then that blocks the ribosome from moving and making a protein. So this is called post-transcriptional gene control, which we'll talk about again later. All right, some background before we talk about the mechanisms of polypeptide synthesis. We've got the tRNAs. So what's going to happen with the tRNAs? 80 nucleotides long, a little bit of double bonding between the chains, giving it this kind of upside down L shape. And at the end, you've got the 3' end, and this is where an amino acid will be attached to bring it into the ribosome. So this is your amino acid attachment site. And then at the other end, down here, you've got this part that's called the anticodon. So remember that our mRNA, the messenger RNA, has the triplet codon, those 3 nucleotides. So if it was UUU, remembering that in RNA we have uracil instead of thymine. So if it is UUU, it will match to this AA, well, that's a G, I site. So just pretend this is an A right here. So AAA on the tRNA would match to UUU on the mRNA. That's how they know to match. And then this particular tRNA always binds to a particular amino acid. So if it's UUU, that matches to AAA, which matches to this particular amino acid. And that brings the correct amino acid into the ribosome to build the protein. Okay, so what we see then is the 3' end is attaching to that amino acid. That's going to be specific to whatever tRNA it is. And then your anticodon is on the other end right here. And that is matching to the 3 nucleotide codon section of the mRNA. So the DNA is called the code, the mRNA is called the codon, and then the tRNA is called the anticodon. And if you were to look at this, if the mRNA is AUG, then the DNA would have been TAC. So TAC is almost identical to UAC, except we have replaced the uracil with thymine. So the anticodon is the same as the DNA because everything is anti-parallel and complementary to the other one. So the way this all works, of course, it's going to be enzymes. So what we see here, this enzyme is called tRNA aminoacyl synthetase. No, you don't need to know that. It's just the enzyme that puts the amino acid together with the tRNA. That requires some ATP, of course. So this binds, and so you've got one of these enzymes putting all of these things together, and this has to happen before polypeptide synthesis. So the tRNA has to already be attached to that amino acid. So there are 45 different tRNAs. There's only 25 amino acids. So that means some of the tRNAs are going to carry in the same amino acid as another one. And so this is important because this is what's known as the wobble effect. So a lot of our mRNA codons will bring in the same amino acid because the third letter of the codon, so if it's AAU or AAA, they may bring in the same thing. So that minor mutation may have no effect whatsoever, and this is what's known as a silent mutation. We'll talk about that later as well. So typically, it's the first 2 nucleotides of your codon that are most important, and the third might be important or it might not be. All right, so another player in the game is the ribosome. The ribosome is made up of 2 parts, the large subunit and the small subunit. Both of those are made up of RNA and protein. They are assembled in the nucleolus inside of the nucleus. And the small subunit is going to bind to the mRNA. And then the large subunit comes into place. And then these 3 sites, the A site, the P site, and the E site, are where the synthesis process happens. So the A site is called the amino acyl site because that is where the amino acid comes in. And then the P is called the peptidyl site because that's where the growing polypeptide chain is. And then the E site is the exit site where the empty tRNA leaves the ribosome to make room for the ribosome to slide down the mRNA and the next tRNA come in. When we look at prokaryotic and eukaryotic ribosomes, there are some differences. So we can see that in the prokaryotic ribosome, there is the small subunit is called the 16S subunit. And then the eukaryote is called the 18S subunit. And that's important because that makes them different. That allows antibiotics to affect them. And so we can kill bacteria with antibiotics because it targets their ribosomes, but it does not target ours because they are structurally different. In a lot of DNA analysis, you will either look for RNA, not RNA, you will look for ribosomal genes, either the 16S gene or the 18S gene. If you are looking at the 16S gene, you are looking at prokaryotes. If you're looking at the 18S gene, you are looking at eukaryotes. And you can use that to identify species and to trace evolutionary lineages. And we'll talk about that when we talk about DNA technology and when we talk about evolution. All right, so again, DNA, this is our code that is copied anti-parallel to the codon, and then matching this would be your tRNA, so that'd be A, C, C on the anti-codon, which would then bring in this tryptophan amino acid. So in transcription, this is where we're going from DNA to RNA. And so the template strand, the 3' to 5' strand, is going to be what is copied into the RNA. So the other side of the DNA, the part that's 5' to 3', is not read. And what tells the cell where to start are going to be what are called promoter regions before the DNA, which is called the transcription unit. So the part that's being copied into RNA is the transcription unit. Preceding that is a promoter region. Okay, so the DNA that is going to be copied into RNA has several different regions. It has the promoter region, which is made up of two things, the Tata box and the start point. So this is the region right before the transcription unit. This is the part that is actually copied into RNA. This is the part that tells the RNA polymerase where to bind. And then this is where it actually starts to transcribe. At the end of the transcription unit is called a terminator region. That's where the RNA polymerase knows where to stop copying the DNA into RNA. And these things right here are going to be controlled by lots of different things. Some of those things we'll talk about specifically when we talk about gene regulation and control. All right, so transcription happens in 3 steps: initiation, elongation, and termination. Translation will also happen in 3 steps: initiation, elongation, and termination, because scientists are not clever in naming things. And so moving on. All right, so in the initiation process, the Tata box, a series of thymine, adenine, thymine, adenine, thymine, adenines, that is going to be bonded to by a series of proteins called transcription factors, which you need transcription factors to allow transcription, but transcription makes proteins, and if transcription factors are proteins, where did those proteins come from? Do they have their own transcription factors? Yes. So going backwards, back, where did the first transcription factors come from in a cell? Again, from your mother and her egg cell, passing those transcription factors down to the zygote, so it has enough transcription factors to start its own process. All right. These transcription factors bind to the Tata box. in the promoter region. The RNA polymerase is then going to binds at the start point, and then it does the job of helicase and polymerase. So it's going to unwind and unzip the DNA and then start copying the DNA. And it copies the DNA 3 to 5, building the RNA 5 to 3. So looking at this picture, this is our transcription unit down here, what we're going to copy into RNA. We've got the top box up here. We've got all of these transcription factors binding. So this is kind of like the guy at the airport or woman at the airport with the flashlights guiding the plane in, like over here, over here. And this is the airplane coming in. It lands, and then it's going to start unwinding the DNA, and it's going to copy that into RNA. This is helped by different enhancer sequences upstream from the DNA, which are activated by proteins. And then these activated enhancer sequences guide in the transcription factors. So we need proteins to bind the DNA to help bring in the transcription factors, to help bring in the RNA polymerase so that it can be copied, so it can do its job and make RNA. So it's super complicated. So this is, again, so this is our transcription unit right here. This is our promoter. And so way upstream on the DNA, way up here, there are these enhancer sequences that activators bind to, and these activators bend the DNA and they guide in all of these transcription factors, and then we can start transcription. So the takeaway from that, this is about gene regulation and gene control. Your cells aren't always making all the proteins all the time. Sometimes your cell needs to make proteins and sometimes it doesn't. So sometimes your cell has a high level of these activators, which then cause this gene to be expressed. When we don't need this gene to be expressed, we can have repressors that will bind to the enhancer sequence, and then it stops this process. So we have built-in on-off switches so that we don't always make stuff that we don't need. So moving on to how the RNA is actually built. The RNA polymerase goes along the DNA, separates the strands, and then is reading the DNA 3' to 5', building the RNA 5 to 3. And it is built complementary to the DNA. And so if the DNA is G, the RNA will be C. If the DNA is C, the RNA will be G. If the DNA is T, the RNA will be A. But if the DNA is A, instead of T, we're going to have uracil instead of thymine. So again, 3' to 5' is how the DNA is read, and then the RNA is built 5 to 3. So things in the AP test, they like to give you a strand of DNA, and they have it marked the ends with their directional features, 5 to 3 and 3 to 5. And then they ask you to transcribe that sequence. And so you have to know that you have to read the 3' to 5' side and then transcribe it correctly. We will have that on the test coming up, so you can get some practice. Okay, so here's the picture of the process. So your polymerase is moving along your DNA. And so we can see that this is our 3' end, so we're following our strand down. This is our 3' to 5' strand. It is being read, T to A to U, G to C, etc. Once it gets to the end part, it is going to then do the termination sequence. I'm going to pause here for a second, and by pause, I mean pause the video. quickly transcribe this for practice, you get AUG, CGG, blah, 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 blah. This AUG is going to be important. We need to note that AUG is going to be the start of almost every protein. And this is going to bring in the amino acid methionine, sometimes called start methionine because it is at the beginning. All right, at the end, it hits the terminator sequence, and then that causes the polymerase to separate from the DNA, and then you've got this strand of mRNA. If it is bacteria, it is ready to go and go into translation. In fact, bacteria may not even finish transcription before translation starts. But if it's a eukaryote, then we go to the next phase, which is RNA processing. Okay, picture of the process again. These videos are good to watch. We will probably watch them in class, and I'll point out things. But if you go on to the topics page, there will be a list of these videos to watch as well. Good to watch. Visuals help. All right, so then RNA processing, and this is another step in genomic control and making sure that the RNA is ready to go. In eukaryotes, we have a lot of sequences of DNA that don't actually code for proteins. So they get mixed in with our coding sequences. So part of this process is to remove the non-coding sections and get it ready for actual translation. So this is what's known as pre-mRNA. This is your rough draft. So you write a paper for your English teacher and you turn it in, and then your teacher's going to say, you need an introductory paragraph and you need a concluding paragraph. And then in this middle, you've got all this junk that just doesn't belong. Get rid of it. That's what happens with RNA processing. So we're going to modify the ends, and then we're going to remove introns, and we're going to splice exons. So I said we had a lot of DNA that doesn't get coded into proteins. That's what is called an intron. And then the exons are the ones that are expressed as proteins. So exon expression turned into proteins. Introns, think of as interrupters. And if they were left in, they would break up the information that the exons are trying to make. So there's lots of theories about introns. Why are they there? Some scientists think that they are leftover pieces of DNA from viruses that have inserted themselves into our DNA over the evolutionary timeline. Some scientists think, and this is more the kind of favored theory, that they're starting to help control gene expression, so they might have some actual function, but they just aren't turned into amino acids and into proteins. All right, so on the ends, we have a 5' end of our mRNA, and we've got a 3' end. On the 5' end, we are going to add a guanosine triphosphate, a GTP. This has two functions. It is going to help bind the mRNA to the ribosome, and it is going to, that's the second one right there, and it also helps prevent the mRNA from falling apart. So this is one of the big differences between prokaryotic and eukaryotic mRNA. Prokaryotic mRNA, the bacteria, they don't do this. They don't put these end things on the mRNA, so their mRNA breaks down very, very quickly. So they have to constantly make mRNA. Whereas eukaryotic mRNA, because it has this 5' cap, or the GTP, and then on the other end, you have a poly-A tail, lots and lots of adenines, like 200 adenines long. The poly-A tail and that 5' end, they help protect the mRNA so it can last for hours, days, weeks, or even longer, so that our cells don't have to keep using up resources to build RNA. So, modifying the end, add a guanosine triphosphate 5' cap. So it can be called the GTP cap, called the 5' cap. And then we get the 3' end, which is called the poly-A tail or the polyadenylated tail, which is just a long chain of adenines. Okay, so that's taking care of the ends. All the stuff in the middle is where we need to be doing our gene editing. And that is where, okay, stop, 5' cap, poly tail. And then in the middle, we've got lots of nucleotides. That is going to be the introns and the exons. So getting rid of the introns. So the introns are in the DNA. The exons are in the DNA. They are copied into the mRNA. So we still call them introns and exons, but they are in both places. We need to get rid of those introns because if you leave those in there, you get a whole series of extra amino acids, which will then change the primary structure, the secondary structure, the tertiary structure, and then you'll have the wrong shape, wrong function. That protein won't work. That's going to be an issue for when we talk about putting eukaryotic genes into prokaryotic organisms, because prokaryotes don't remove introns and splice exons together. So when we take an eukaryotic gene and we put it in a prokaryote, we have to take out those introns to make sure that the bacteria makes the protein that we want. We'll talk about that in our DNA technology unit. All right, so our non-coding DNA, intron, so again, for intervening or interrupting. And then our coded DNA, the exons, the ones that are expressed into proteins. So every cell that is working is really only expressing 1 to 2% of its genome. A lot of it is intron DNA, some of it's for RNAs and other things, and there's just a lot of DNA we're like, we don't quite know what that does yet. So again, this technology is only 70 years old. So there's a lot about DNA and its control that we're still learning. All right, so here is a pre-mRNA sequence. We have exons. We want to keep those. And then these introns we don't want. So the cell is going to cut the introns out. It's going to splice these together to give us our final form down here. This is controlled and regulated by those small nuclear ribonucleoproteins, or these SNRPs. These cluster together with proteins to make a spliceosome, and these come in and they fold up the mRNA, cut out the intron, and splice the exons together in one kind of swift motion. And that's going to give us our final mRNA. So this cuts out the introns, attaches the ends of the exons, and it looks something like this. So you have all of these SNRP proteins and RNAs, and they come in, and they fold up the intron, and then they're bringing the exon ends together. And so then this intron mRNA gets broken down and recycled. So here again, this is everything together as the spliceosome. And then we have our final mRNA. We are ready to move on to translation. So again, some of these videos you want to watch to help visualize that process. All right, translation is really where we are building the protein. So this is going to require our final mRNA. And it's going to require the tRNAs with the amino acids that are attached. So your basics, mRNA attaches to the small subunit of the ribosome. tRNAs match up to the mRNA and bring in the amino acids, and then it builds the amino acid chain. And then the polypeptide detaches. It is not that simple, but it's also not super hard to understand. It is this picture, so if you can memorize what's going on in this picture, you understand the whole process. So you've got your mRNA right here. It is sandwiched in between the large subunit and the small subunit. And what happens, we've got that codon sequence. So we've got the like an A, a C, and a G right here. Pretend that's what that says. I can't see it. And so this tRNA is matching that A, C, and G with a U, G, and C, and it brings in this specific amino acid. Well, this is our growing protein chain. So this site of the ribosome is open. That's the A site. So a new tRNA comes in. The anticodon here is matching to the codon here, slides in, and then this is going to bump over onto the top of this. This becomes empty, and then we'll go into the E site and exit like this one is doing here. And then this one, which is in the A site, will move down to the P site, and then the A site will be open again. Super simple. And then it just keeps doing that until it's over. Your detailed steps, don't worry, we'll go over that in class, is, again, okay, so the beginning of translation is initiation. And in this process, the 5' cap, that guanosine triphosphate, is going to attach the mRNA to the small subunit of the ribosome. And then the first amino acid is going to come in on its appropriate tRNA. So on the mRNA, the first codon sequence is AUG. So we remember that the first codon is August because we start school in August. This is called start methionine. And that's going to be the amino acid that is in at the beginning of almost every single protein. So then what happens once this attaches, then the large subunit kind of slides in. So this first tRNA is going to be in the P site. And then we're ready to start building. So this is what we see here. We see that our the end of our mRNA is going to bind to our small subunit right here. And then, oh, hi, buddy. You want to come say hello? Come on. Say hello to everyone. Pabu loves protein synthesis. Don't you, Pabu? Yes, you do. Ugh. No, I don't want to see your butt. All right. So the first tRNA, we were matching to AUG right here. We are bringing in start methionine, and then that large subunit slides in so that the tRNA is in the P site right there. So then over here is the E site, here's the A site, and then we're ready to start building, and we start building by bringing in the next tRNA into the A site. So now think about methionine. It can show up in other places in the protein. It's not just the first one. It can be other places. So we only call it start methionine when it's the first one. When it shows up later, we just call it methionine. All right. So elongation is when we start building the polypeptide. This happens with codon recognition. This is where the anticodon of the tRNA matches to the codon of the mRNA, and that brings in the next amino acid into the A site. So we have these two charts to determine what amino acid comes in. This is called the codon chart because we look at the codon. So if we look at this one, the easy one, so we see if the codon is AUG, this is our first A to U to G, and that's methionine. If it is GCC, G is the first one, C, and then C is alanine. And we can see that here, GCU, GCA, GCG, GCC, they all bring in alanine. The thing that's important is the G and the C. That's what's known as, that's that wobble effect, where if you have any one of these, you're going to get the same thing. It's not true. So if we look at U, like UC whatever gives you serine, but UAUG gives you tyrosine, UAAG gives you a stop codon. So it's not just the first two that are always important. Sometimes the third one is important. But sometimes over here, as you see, it's not. This nice, easy one usually does not show up on the AP test. What they give you is this one over here. And so this is your first letter. This is your second letter. And then this is your third letter over here. And so if it is AUG, so we see that, so if this is our first letter here, we go to this row. And then at U, this is our second one, so we're in this column, so that would put us in this square. And then the G is over here, and then you can see AUG gives you methionine. So if it was CCC, first one right here, second one in this column, and so we're in this grid, and then CCC gives you PRO or proline. You don't have to know the names of any of these except for methionine. On the test, you can just write these 3-letter abbreviations for each amino acid. So we're going to pause. I'm going to say if you were given, we're not going to use this one. We're going to use the harder one. If we did GGG, what would that give you? Except you can't see it on the screen because my head is in the way. So let's try UGG. And so we see that U is here, G is here, and then UGG gives you TRP, which is tryptophan. We will practice that in class. Okay, so pause the video for a minute, and then you want to look at your mRNA. Every 3 letters is your codon, and you will. And what I want you to do is to transcribe that into, not transcribe it, translate that into your amino acid sequence. So we know that AUG gives us methionine. Then CGG would be CGG, which is ARG, arginine. And then keep doing that for the rest of them. And we'll check over that in class. All right, so the next step of elongation, we have our original start methionine in the P site, and then next to that, we've got the next amino acid in the A site. And so the P site amino acid will get bonded to the top of the amino acid in the A site. And so we're forming a peptide bond between the 2 amino acids. And then we get the process of translocation. And this is where, as the ribosome shifts down the mRNA, the tRNA in the P site goes to the E site and leaves, and at the same time, the tRNA in the A site goes to the P site, and then our A site is open again. And then a new tRNA can come in, and then we'll shift the amino acids from the P site to the A site. We'll translocate again. A site's open, new tRNA, new peptide bond. And then we just keep going and going and going and going. We'll definitely go over that in class. Watching the videos on that will help as well. And then this is going to continue until you get to the end of the mRNA. And at the end of the mRNA, there needs to be a signal to stop the process. And the process of stopping will be the termination sequence. And that's going to require what's known as a stop codon. All right, so let's look at this process. So here is our growing polypeptide chain, or if it's just one, that would have been our start methionine. So we have our codon recognition process where the tRNA comes in, it binds, and then our protein chain is going to shift over onto top here. We see that. And then the translocation, so this is going to go to the E site. This will then go to the P site. and then our A site is open again, and we just keep doing that over and over and over, and this protein chain just grows and grows and grows until we reach a stop codon. So there are 4 codons you need to know. AUG, start codon, brings in methionine. And then you need UAA, UGA, and UAG. These are your stop codons that stop the process. So these bring in a protein, which is called a release factor. And instead of having amino acid, when the ribosome shifts the polypeptide to the A site, there's no amino acid to form a peptide bond. And so then it gets released by the release factor and everything falls apart. And then it's done. And then your protein needs to go from primary to secondary, tertiary to quaternary with the help of chaperone proteins. And your ribosome breaks apart in the large subunit and the small subunit, and then it goes and binds to another mRNA. And then your mRNA can just bind to another ribosome and make another copy of this protein. Release factor comes in. This goes to bind to an amino acid, but there's nobody there. So then it goes off into the cytoplasm or into the lumen or inside of the rough ER, and then everything disassembles. Good video to watch to help you understand that process. Now, one thing that can happen is what's known as polyribosomal action. So basically what's happening is here's your mRNA, and you've got lots of ribosomes on all at the same time. So as one ribosome slides down, it leaves space for another ribosome to bond. And so you can have lots of ribosomes on a single RNA, making multiple copies of that protein all at once. If it is a prokaryote, what we can see is that the DNA is being copied into RNA, and as soon as the RNA is coming off the DNA, ribosomes start binding to it. And then even before the RNA is done, you have all of these ribosomes on the RNA making lots and lots of proteins, because those things are small and they have to grow fast. So they start translation before transcription is done. Whereas eukaryotes, you have to finish transcription, do your RNA processing. But once that's done, then you can have multiple ribosomes attached, making lots of proteins all at the same time. Okay, so here we see the transcription and translation of, in bacteria or prokaryotes, happening at the same time. Got your polyribosome chain. Transcription, RNA processing happening in the nucleus of the eukaryote. Then the finished RNA comes out, and then you can have multiple ribosomes attached, building your protein. So then your finished products, cytosolic ribosomes. These are the ribosomes that make the proteins for the cytoplasm. Oh, hi. Did you want to say hello too? This is Milo. Say hello. Say hello. No, I'm Milo. I'm Mr. Irvin's favorite because I'm handsome and fluffy. Okay, so cytosolic ribosomes. Cat hair. The bane of my existence. Cytosolic ribosomes make proteins for the cytoplasm, and so there are chaperone proteins. Oh, what's going on? You want to sit on my lap? And so the chaperone proteins are in the cytoplasm. So after the proteins are made, then they will get their final shape to do their job. There are lots of post-translational modifications to give them their correct shape, but cytosolic ribosomes are going to make those proteins that the cytoplasm needs, like for glycolysis and running the start of cell respiration. Bound ribosomes are the ribosomes that are going to make the proteins that go into the rough ER. They're called bound because they bind to the rough ER, but they start out as cytosolic ribosomes. But these are the ones that move to the surface of the rough ER based on that signal peptide that's coming off the beginning of the protein, and because of that signal recognition particle RNA that leads it to the rough ER. So this is that process again. So you've got a cytoplasmic ribosome. The protein is destined for the ER, so it binds to this RNA, brings it down to the rough ER pore that opens up, and then the protein can enter in. What, buddy? It can enter into the rough ER to be secreted from the cell. Okay, so what happens if our DNA changes? So change the DNA, change the protein, right? Maybe, maybe not. So we've talked about lots of chromosomal mutations before with aneuploiding and errors of crossing over. So these are the chromosomal mutations. So you get a whole chromosome gain or loss with trisomy and monosomy, or you can have deletions, wrong crossover things. And so that could have major consequences because you've got extra genes being copied or lost. Hey, what are you doing? You're going to mess things up. So, and then you are going to have lots of problems in those cells. You can have errors in DNA replication, and these typically result in what is known as a point mutation, where one nucleotide difference happens because it was transcribed incorrectly, and then the correction enzymes did not catch the error. So we saw this in sickle cell anemia. So your normal DNA is CTT. That was mutated into CAT. Normal DNA brings in a glutamine because CTT is made into the RNA GAA. CAT is made into GUA, and so GUA brings in valine. This is a polar amino acid. This is a non-polar amino acid. So that causes the protein to change its shape, which gives you all the effects of sickle cell anemia. So point mutation. So if we're looking at this, we've got the wild hemoglobin, and then this is your mutant hemoglobin, CTT to GAA to GUA, valine instead of glutamine. Now this is what's known as a missense mutation, where the protein that is made is actually incorrect. But this is not the only type of point mutation. So the other types include a silent mutation. So this is where you have the wobble effect, basically protecting the individual from the mutation. So this is where if CCG is changed into CCA, then your resulting RNAs are GGC and GGU. Well, if we look on our chart over here, right where my head is, they bring in the same amino acid. So because even though there was a mutation, the end result is the same amino acid. So nothing happens. So that's why it's called a silent mutation, because you would not know that this happened unless you did a deep genetic analysis. The next is going to be so here's our wild type. Here's our mutant type, both bringing in glycine. No problems. Okay, so missense mutations, again, this is sickle cell anemia. So this is where the DNA mutation causes a different amino acid to come in. And that probably will cause an issue, but it might not. So with sickle cell anemia, we're replacing a polar amino acid with a non-polar amino acid. So they are chemically different. So that's going to cause issues. But if you have a polar amino acid replaced with another polar amino acid, or a non-polar amino acid replaced with another non-polar amino acid, it might not have enough of a difference to have a large effect. So sometimes this could be bad, sometimes it might be fine. All right, the next is going to be what is known as a nonsense mutation. And this is where you have a stop codon being brought in earlier in the protein, so the protein doesn't get finished. So this happens when you have something like AGA as your mRNA being changed into UGA, and then that would bring in a release factor. All right, so if we look here, we can see that our wild type is AAG, which would bring in a lysine. But down here, the first part of the codon is changed into UAG, and so that brings in a stop codon. So that means all of these amino acids that should have been put in are not going to be put in at all, and then that protein is going to be completely non-functional. So that could have huge devastating effects on the organism. But again, it depends because we are diploid organisms and we've got 2 copies of every gene, at least 2 copies of every gene. If this is the bad one, then this one might make enough of the enzyme or protein to make up for it, and therefore you still show the dominant trait. And that's why typically you don't see genetic diseases being necessarily harmful unless it's in the homozygous recessive condition. All right, something that typically almost always causes an issue is something known as a frame shift mutation. And this is where you are either inserting one or more nucleotides into the DNA, or you are deleting one or more nucleotides. And so this causes a shift in what is known as the reading frame of your mRNA. So in your DNA, let's say that you have 12 letters in your sequence. Well, due to crossover error, you lose one of them. So now you're down to 11. Well, you read every 3 letters. So if you've got 12 letters and you lose the second one, well, instead of reading 1, 2, 3, you're now going to read 1, 3, 4. Instead of 4, 5, 6, you would read 5, 6, 7. And so everything just shifts down by 1 nucleotide, if that was a deletion, or one gets inserted, and then everything shifts the other way, and you go from 12 bases to 13 bases, and then everything is just off kilter a little bit. So what we see here is you've got this whole sequence, and then we've got an insertion right here. So this should have been read UUG, but then it's going to be read UUU, and then GGC, and then UA. And so it just shifts down, and a deletion would just like removing one, and then it shifts the other way. And so after that insertion or deletion, everything after it is going to be different. So that causes huge issues with that protein, unless you insert or delete a triplet, and then you just basically skip 1 amino acid, or you add 1 amino acid. And then potentially that could have a problem, or it might not have any problem, depending on what amino acid is gained or lost. So then deletion, and so both of these insertion and deletion, everything gets shifted, and sometimes you'll end up having the shift cause a premature stop codon to happen, and then that cuts your protein off even sooner. All right, stuff to watch, crash course videos, good stuff to help you see it. Hear my best friend Hank Green talk about it. And then we will go over this in class rapidly again, so please take your notes and listen so we can go through it quickly in class. All right, I hope you had a good couple extra snow days and that everything is good at your house. All right, see ya.